With this introduction, so I would like to jump to my presentation. So my today's presentation, okay, can be entitled as, just a minute, my pointer. Okay, so it's a trend in pharmaceutical industry, or importance of material science concept and advanced analytical tools for co-crystal developments. Okay, so with this, okay, so just I would like to give a brief idea about my outlines. Okay, so today I'll try to cover mostly 6 topics. Okay, so that's the last 2 topics includes the case study. So that's our recent publications. Okay, and before that, the first topic. So I would like to give a uh, some impression about how the pharmaceutical industry is doing, where it's moving, where it's going ahead. This kind of thing I'll discuss in the first topics. And second thing, so I will try to put my arguments or put forward why this multi-component API is difficult and why it can be considered as a complex drugs. Okay, I'll give some arguments. Okay, and then to handle this what Ramanaya also discussed very nicely that what are the way forward, because the industry nobody want to listen that is difficult. If there is a business, people will be expecting that yes, you have to do by hook and crook. So what are the things we can approach, or we can take it way forward? So in this concept, so I'll be introducing the pharmaceutical material science approach, how it can help, and and also same time the advanced analytical tools, how it can help. Okay, so mostly I have tried to estimate all these topics. So probably I'll be talking around 40 minutes or max 45 minutes, and then we have a discussion question and all this. Okay, so let's start with what is pharmaceutical industry. Okay, so it's a very general, but I will put my some highlighting points. Maybe we will converge somewhere. Okay, so yeah, so it's, all pharmaceutical industry is a part of healthcare sectors, and that deals with the medication. So we make medicines. Okay, uh, and if you think in a bit broad way, okay, pharmaceutical companies can be of 3 types. Okay, first category, they only make APIs or KSMs. Okay, means intermediates. Okay, they're practically only a chemical companies. Okay, and like a example, you can take is a Debris Lab or RT drugs. Okay, and second category is the generic drugs, drug players. Okay, so as for example, yeah, Dr. Reddy's, Lupin, about laboratories, Smile and Sun, yeah, all these things. So what they do? So they take the off-patent drugs, okay, and they try to formulate and they try to market it, okay. And third category, it's the biggest category. It's the innovation, real innovation is happening there. That's the innovator, okay. They make the patented drugs, okay. And as for example, you can take Novartis, Pfizer, GSK, Teva, etc. So if you see all these pharmaceutical this space, it's really growing economy. Okay, it had been estimated that by 2023, so this globally this industry will be reaching 1.5 trillion. Means there is a lot of opportunity and all these things. So with this context, so I want to give you a idea. So what is happening in happening in a pharmaceutical industry, where we are approaching. Okay, so there are few trends. Okay, so first trends you will see. So yeah, there is a really big hike about the big data. Okay, AI, ML, and they are going to be used as a preventive or predictive measures. So starting from the drug developments. Okay, so all we know the how the repurposing of drug is coming up, and this nothing but it's from the data mining and using the old data, old information, and then taking some leads and doing forward. And same time, this technology also going to fix a marketing strategy also. Okay, that's how the Amazon is doing and all these things. So pharma also moving in towards that thing to make a big impact on the markets and all these things. Okay, second trend I'll be talking. So it's a digitalization and continuous manufacturing. This technology it going to enable the pharma 4.0. So it's nothing but that all the plants would be should be digitalized, they're automated, and then manufacturing will be much more smooth. It's a dream. That's the thing is, it's happening. All company taking that right. Okay. Okay, about the third thing I want I want to highlight. So it's a regulatory environments. Okay, so now is regulatory environment is really favorable. Okay, of course they are for innovator. Of course they are for generic, because generic company can make a affordable medicine. Okay, and they can create a continuous support to the overall world. Okay, so in that sense, they are really helping the generics players, okay, to make a product specific guidance. Okay, they are really entertain the any under players for a pre under meetings to give a scientific thoughts, to exchange their scientific thoughts and way forward how they can make their product in a reliable and faster manner. Okay, so in a topics 3, 4, 4, 5, 6, they are interconnected thing. Here is the main thing is happening, the biotechnology and nanotechnology. Okay, so the different drugs are getting hiked. Okay, and they are targeting for curative medicine like a gene therapy or cell therapy, or also trying the complex drug and biosimilars. Yeah, and same time, so if you see, another big trend is happening is a merger and acquisition. If you see, okay, so there is a lot of new biotechnology, nanotechnological companies. So innovators, spin up company innovator it's acquiring. Okay, and same time, if you see that that's another big trend in change in the last couple of years. Okay, innovator is merging with the generic companies. Okay, this effort is nothing but the increasing the inter organization collaboration. So at the end, what it's targeting? So all it's targeting is delivering precise, innovative medicine in a faster and uninterrupted manner. So please note that this uninterrupted manner and faster is really important. So now I think after last couple of years, swimming through the all COVID situation, all we understand how much is needed. It has to be a drugs, has to be in a faster and uninterrupted manner. But otherwise there is a lot of deaths and all these things. So that's not very difficult to understand in a current situation. Okay. With that, so I'll just as I given the introduction. So I will discuss about the why the multi-components API can be considered as a complex drugs. Okay. Before that, I would like to give an idea about what is complex drug. Okay, so complex drug, okay, can be of 4 category. Okay, so first category is nothing but it is a API itself is a complex. It could be a peptide based drug. It could be a polymeric drug, or it could be a complex drug of API. Okay. And second category, if you see that there is it is a formulation is difficult. Okay, so in terms of example, you can take it as a liposomal drug, emulsion drug, gel, and colloids. Okay, and third category, if you see that this is a way of delivery is different, innovative, as for example, dermatologicals or the inhalation drugs. Okay, and in a fourth category, it's a device. It is an innovative device design. So, for example, auto injection. It's really a game changer. If you think that for taking an injection, practically for the diabetes people, okay, taking an injection and going to the hospital is very tedious. Now innovator company came up with the auto injection, so you can take patient can take their injection by themselves. It's a really fantastic innovation. Okay, now if you think that any complex drug can be combination of all of these or some of these. So in a different way, it can be told that each complex drug, it's complex in its own way. Now, to connect that complex drug and multi-component, so I would like to present a paper, very recent paper by E.J. Myers, and also Ramanaya also was talking in the same sense. That's also I am talking with his from experience. I'm talking from the paper. They are converging. So let's go through that. Okay. So this paper they are talking about the construction of supramolecular synthesis. So in this context, that the few points I would like to highlight. Okay, that will give an idea where we are and why the multi-component drug is not easy cake for industry. Okay, so there is a paradigm shift of covalent chemistry to non-covalent synthesis. Okay, so we know their supramolecular synthesis is very much needed for, is not for the pharma. It's so many other industry, so many good functional material can be synthesized. Okay, so if you to look in a reality for synthesis of large molecule with a multi-step is not, it is still is not a more challenge. But if you think about of a synthesis of a non-covalent synthesis in a reproducible, in scalable manner, it's we are practically in an infancy. We have to put a lot of efforts for this. Why that thing? So that can be answered in a very simply way. Okay, such systems are dynamics. Okay, and emergence in nature due to the competition of various kind of interaction. As Ramaiah told that it somewhat is driven by the hydrogen bonding or some weak interactions. That controlling is not so much easy. Okay, and so there is a level of uncertainty and difficulty. It's so much that is controlling is really difficult. Now, on the other hand, if you look from the perspective of multi-components APIs, okay, so where co-self assembly, okay, means selective molecular recognition overrule the self assembly, because all we know self assembly is a much more favorable naturally and all these things. So here we are going against nature, so we want to make co-self assembly. Okay, so yeah, so there are that's the difficulty comes. Okay, of course that there are subtle thermodynamics and kinetics interplay. Okay, creates difficulty in selection process and of course in a scale of control. Okay, and the next stage is the in a formulation stage, the excipient compatibility. Okay, and formulation process tolerance. So with having a co-crystal is not enough unless we are make it as a finished product, as a capsule, tablets, injection, whatever. That the real challenges come for the multi-components or co-crystal systems. Okay, with that I would like to give some idea about the marketed drugs. So as Ramaiah told that yes, there are some innovator company, they are successful, but as a generic players, we are struggling still. Okay, so but here I don't go to the list, but I want to highlight a drug. Okay, it's the instance stroke. Okay, it is called also LCZ695. Okay, it's by a Novartis. Okay, it's a drug complex of sacubitril and valsartan, and it's used for the heart failure. And if you look the structures, you will be amazed how difficult can be a structure. Okay, so if you look the asymmetric of the crystals. Okay, so it's a one, it is a drug complex of valsartan and sacubitril, but there is that is mediated by the 3 sodium ions and 2.5 molecule of water. So it's a very complex molecule, the structure, let's say. Okay, and then and authors, okay, by the Novartis, they called it as a supramolecular complex, because if you think in a landscape, so it's everything. So salts are there, hydrates are there, co-crystals are there. So that's why it's very difficult to. So everything existing there. In the next slides also you'll be get it. Okay. So yeah. So it's a practically monovalent multi-coordinated sodium cations and water molecules. Okay, and see the unit cell. It's so big. It's a really big supercell. Okay. And same time, so these molecules are also having a solid state forms. Okay, there is form 1, form 2, also coamorphous. And what is more surprised me that it's when the coamorphous developed. Okay, so coamorphous also share the same local structure of crystal structures. So that is being a so complex but structural, there is some connections and all these things exist. Okay, that's a really surprising for me. Yeah, that's the thing. Okay, with that, so now let's review the landscape of solid state form of drug molecule as a material. Okay, so it's a very systematic routes to fine tune the pharmaceutical or mechanical property. Hence we could reach a better manufacturability. So if you, it's a very common diagrams. Okay, if you see, what I would like to highlight, that's multi components give us more opportunity. being a salt, solid dispersion, solvates, co-crystals. Co-crystal can be also different kind. That's also I'll also discuss one new more category we feel can be another category can be a subsets. Okay, and also the coamorphous. So there is much more possibilities is there if you end up exhausted with a single components like a polymorph of amorphous. But there is a difficulty, there is a very strict checkpoints kept by the agencies as for example. So stoichiometrical characterization has to be very much confirmed. That means crystal lattice, you need to have a atomic level or molecular level structures. Another important aspect is the dissociability of multi-components material before absorption is mandatory, because drug always in common it's a drugs play their role with interacting with a some protein and changing the signaling path. So before getting to the targets, it has to be dissociated. Otherwise, total game is wrong. Okay, so that's is a very difficult. That's why the co-crystal agency will be looking for much more data. It cannot be just a dissolution data and this thing is cannot be. They will be looking for a lot of data. Okay, with from this perspective. Okay, then the third category is so if you have a salt or co-crystal, the absence of ionic interaction to be established by the spectroscopic or other orthogonal technique for co-crystals or salt. So you should have a very strong solid state characterizations to confirm all these things. So that's also not enough. So you have to have the so many very high end the solution characterization also as a molecule, as an ion pair, or as a cluster. So whatever you can call it. So more. With that, so I am going to introduce about the analytical tools. Already in Ramaya also presented that thing. So I'm not going to very details on this thing because it's very known. Okay, and what I would like to highlight, okay, is following. So here this set of techniques can confirm the molecular level same time. Okay, so also the bulk level, like for a molecule particle size distribution is really important. Okay, hygroscopic behavior is really important. Okay, and its rheological behavior is very important factor for the process. All these thing has to be combined. As a microscope, its morphology has to be understood to interpret all these things. Okay, so in a combination, so that's all these things we needed for as from solid state perspective. But that's not all. So you have to have the separation technique like a LC to make sure about the impurity profiles, and same time the dissolution technology to make sure that its molecule also dissolving in a favorable way. And but and few things I want to highlight. So some new updates whatever the having we have. So one is the single crystal. All we know single crystals is the gateway to get the molecular length scale. Okay, it's a molecule how it's what is doing in a three-dimensional, how supramolecular is interacting. So conventional technique is the same single crystal XRD. Okay, but sometimes getting a single crystal is difficult. Then people found out that yes, synchrotron can be a way out. Okay, so now people, yeah, here actually you can work with a much more smaller, some micron level crystal. Now people, that's also sometimes get difficult. Then people find out by 3D electron diffraction, so you can really work with a powder. Means you no need to spend any time on your going a single crystal, because inside a powder you will be getting a 100-200 nanometers crystals, and they are more than enough to have a atomic structure. Okay, and now really and very in a last week only this instrument is launched in a IUCR. Okay, yeah. That's a really upcoming technique, and I believe that will be it will be a game changer. Okay, okay, same way. So there are the so many other hyphenated orthogonal techniques also needed. For example, let's consider the Raman imaging. Okay, so Raman imaging. So morning also you have remember. So how the Samir was showing that how that it can use as a in process in situ probe. Okay, and here what I am talking, it's another aspect of the Raman. It's a really versatile tool. Okay, and here I am talking in a solid state, how you can, by chemical imaging, how you can make sure about how your API is doing there in presence of excipients, especially for the finished product. How it interacting with the excipients, is it still is in the same polymorph, is converted or is dissociated, and so many information we can get it from the Raman imaging. Okay, same time, okay, some IP related technique like a PLM, SEM with a Raman technique, so we can see each particles and we can map it what is that particles. Okay, and sometimes solid state NMR also give a really lot of information to us. I'll give some example of these things. Okay, and another category is the time resolved study. Okay, so for time resolved study, for so all we understand from the Ramaya talk that, yeah, we have to understand what is happening in a crystallization. Okay, morning also Swami talked this thing. Okay, so that thing means you have to have a very fast technique to monitor these things. Okay, very fast, because the nucleation growth happens in a, let's say, in a seconds or minutes. Okay, with having a proper supersaturation. Okay, so that's their synchrotron powder XRD, or say or synchrotron small angle can play a big role. Another spectroscopy, that is the time domain terahertz, is coming, is a really big way to monitor all these in situ studies. Okay, so with that, okay, so now I going to introduce about the pharmaceutical material science. Okay, so this is the, yeah, it's material science, but pharmaceutical material science, it's what we are bit different than material science, because here we are dealing with the organic materials, small molecule, peptide, sometimes proteins, sometimes maybe polymers. Okay, they are really soft in nature. They are not hard like inorganic materials. They are less crystalline, amorphosity is more, and all these things. So actually it's a extent of the material science. Technology can be used, but you have to be more thoughtful about your experimental design. Okay, and same time, being an organic molecule, having so many anisotropy and possible of interactions, that's also become difficult. That's already I have discussed about the problem. Okay, it's having a 4 arms. Okay, so that is structure, property, process, and performance. So once you get, so already I think, yeah, Kelvin also in a first day talk, he given some idea about the how tabletability can be used, this thing. Okay, yeah, it's practically, I think Kelvin was one of the introducer for this concept. Yeah, it really helps. Now we can take it forward for the further and so many other trips also. Okay, so structure looks at structure means it could be molecular structure, it could be supramolecular structure, so it could be particle size morphology, as a bulk powder. So it could be for a device, a tablet, it could be a coating for a device. It's all these things structure. So it's starting from the molecule to overall in a device level. Okay. Okay, property. So physicochemical, for all we know, is dissolution, solubility, and all these things. Surface chemistry, mechanical property, moisture content, and rheology. And if you think the process, so there is so many different processes: chemical synthesis, crystallization, milling, granulation, and freeze drying, and all these formulation things. And at the end of the day, so it has to be give a fruitful things in terms of efficacy, safety, manufacturability, stability, and bioavailability. So idea is following. So it's all at the interconnecting informations, interconnecting relations. So that for a complex thing, if we can connect them in a proper way, if we understand, we may have a better control and better manufacturability and a better success in the future. That's only I would bring like that. So the in that terms, so you can talk that yeah, there is different type of characterization is possible. So structure based characterization, like as for example, like say single crystal study, okay, property based. Yeah, it can be how the crystals morphology are there, how what is the tensile strength along the some axis. Okay, so it's like a AFM study. Okay, at the end of the performance, if we making a tablets, how they are happening. So all these things has to be hierarchically viewed and should be connected to the process. Okay, then the success can come. Okay, and what we are trying to say that, so it has to be linked. Okay, and not, it's not at all easy, because a molecule being a difficult, and as we are dealing with a small weak interaction, not with a covalent interaction, and kinetics are different. So it's not an easy game, but yeah, it can be more systematic study, more understanding can give a more success. That's all. So that's only I want to highlight from these things. So crystallization by cascading the elements of structure, property, and performance into the analytical tool can be a promising way to control process outcome. Okay, and at the end we can get the desired molecular multifunctional API. So with this, I'll come to the case study. Sorry, I'll take some more. Okay, so here, we have studied the multi-components materials of agomelatin and phosphoric acid. It's given in a molecular formula. Okay, so you will understand that why it's a safe to call it a multi-component rather than salt and co-crystals. Okay, that will I'll put forward. And what I would highlight here, okay, so if you have a advanced characterization, if you have a very robust computational protocol, okay, and with combining the very strong crystallographic or chemical crystallographic analysis, okay, if you can connect all these things, at the end of the day, you can visualize or you can propose some mechanism of phase transitions, especially the solid state phase transitions, whatever we have observed here. So it is along the salt co-crystal continuum. So that's I go in my thing, you'll be getting that's how we are extracting the information and moving towards understanding the phase transition in terms of mechanistical way and in a turn, so that hopefully is giving more levers to control the material in a scale up and process. Okay, with that, so why this study was taken up? Okay, so yeah, so agomelatin is a novel the antidepressant drugs, okay, is developed by the Sevier in a long ago, okay, and it's a BCS class 2 drug, okay. Dr. Reddy discovered a multi-component API through salt or co-crystal screening, and phosphoric acid was selected on this screening was selected on the basis of by pharmaceutically acceptable excipients, processability, and age of processability and stability, and this information is patented in 2014 in a European patent office. Okay, and what that thing happens. Okay, so and this is the process as per patent. It's a very simple process, looks like dissolving the agomelatin in a solvent like a ethyl acetate, okay, and then adding the phosphoric acid to the solution in a step, and then isolating the, of course, it will get some precipitation upon addition of phosphoric acid, and isolating and then drying the products. So it looks like a very simple thing and it's very easy to do it. So and all we thought that okay, but what happened? So when we started to take the batches, okay, so we start to see the batch to batch variation. So it is a very preliminary powder XRD study. So as you can say that there are such and such peaks, okay, coming in the batches, okay. So from this thing we put forward the hypothesis. It could be a process related, okay. It could be a polymorphic impurity of any existing forms, okay. That's with that thought we take a detailed investigation. So what we came to know, okay, so we use a very simple technology techniques. It's called the powder indexing. So powder indexing is a very powerful technique. It can talk about the phase purity of the material, okay. What from this thing, what we 100% make ensure that this what is the extra peak we are getting in a powder XRD, okay, they are not from agomelatin polymorph, because there are the 3 to 4 different polymorphs also existing for the agomelatins, okay. So they are not part of the agomelatin pure agomelatin polymorphs, okay. So there is a possibility of new crystalline phase. Okay, that understand. So this problem, okay, force us to understand the material, understand material in a full strength, because we have to have a control strategy to make a reproducible batches, okay. So we take a very simple experiment. So we did a DSC and TGA. So what you can see, that there is a phase transition, okay, around 40 to 40 degrees Celsius, okay, just way before melting, okay. And as per TGA, so it's very clear that there is no residual water, residual solvent loss. So that indicates it's a solid state to solid state phase transition. And more surprising thing, it came that when we did a hot stage microscope, so we heated the material gradually, but I am showing the endpoint after that only. Okay, so what we saw, so there is no change of any crystal morphology, okay. Even the birefringent patterns also, it's a very surprise. In general solid state transition happens, okay. They generally it can change the morphology, okay, because it can happen via via resolution and via recrystallization, nothing can happen. And more surprising, there is no change in the birefringent pattern also. Okay, and with this idea we did the variable temperature powder XRD, okay. What we understand, so yeah, whatever we observed in a DSC, it's true. So if we heat the material at 50 degree, we get a new phase, okay. New phase, that's also indexable, that's I explain you afterwards, okay. And what happen if I cool back this material, so immediately nothing is come, but after some time then again the mixtures is coming, okay. So that give a big clue that why the process is having a mixture, okay. And then we did a very simply heat cool DSC study. So what we can see that we say that is around 43 degree, there is a phase transition, and then when you cool it, this material is get crystalline, crystallized, and around 22 degree, okay, 22 degree, okay. So that's why in a powder XRD we can't see the immediately, because our experiment is done at room temperature, but its crystallization temperature is below room temperatures, around 20. So but if you wait some time, so with the time it start to enriching the RH forms, okay. So from this thing, so it's very clearly understand that is a solid state phase transition leads to a new polymorph, that is we call is a high temperature form, okay. And they are anisotropic, and irreversible in nature, okay. And it's recrystallized around 22 degree, okay. That means, so what the control strategy can be taken as a process training. So if we after precipitation of the material, if we vacuum dry at lower temperature, Okay, then we so lower temperature means we are talking about the far below 43 degree, so there won't be any phase transition. So in that sense we can get a reproducible batches, and it's clicked and it's happened. And in that way process was developed, and it's we filed the DMFs and all this. That's the thing. Okay. Okay. And further whatever the study we did it, it's not from the process control and all these things. It's from the scientific curiosity, and to get a much more details understanding about the molecular level structures. So what we did, so all we know, to get a molecular level structure we have to have a crystal structures. Okay, so we have taken a several trial, okay, to grow the single crystal, but we never able to do that. Then we decided that okay, we can solve the crystal structure from powder XRD. Okay, we taken a attempt. Okay, so the low temperature, so room temperature form was data collected in a capillary transmission. Okay, and a high temperature form was collected in a foil transmission. Okay, so yeah, if you can see that we got mostly similar kind of cell dimension and crystal structures also in a larger view. If you see, it looks like a same structures. Okay, I'll discuss in a details afterwards. There are differences. Okay, and and after refinement we got a reasonable R factors. Okay, that's help us to understand, yes, the cell dimension is quite right, but still you are not 100% certain about the atomic positions, especially the hydrogen atom positions. Okay, because your hydrogen position was fixed manually. Okay, and all we know, to talk about the it's a co-crystal or salt, we have to have a correct atomic position of hydrogen atom. So towards these things, we have further worked. Okay, so here you can see that this molecule, okay, so it's a amide. Okay, is not in generally a ionizable group. Okay. And if you look the pK, okay, it's a pK of agomelatin and phosphoric acid is -1. Okay, so means it's near to impossible according to the delta pK rules that it can form salt. Okay, so but there was some some symposium literature was there. They, without publishing crystal structure, they were calling as it's a agglomerating salt. That's trigger us to look in the more details. So towards that we developed a computational methodology. It's got the energy minimization plane wave periodic density function theory. It's actually it is a dispersion correction DFT it's called. Okay, and a FAF program we have used for this thing. And what would look very surprising, so when we have started to optimize the structure, we see for RT, okay, room temperature form, the phosphorus proton is shifted to the amide. Okay, and corresponding that or for that the corresponding resonance structures is also got changed. That's why the CO bond distance, CN bond distance, and all these things got changed. Okay, and we can rely these things because it's a DFT calculation. We can be sure about the atomic bond length and bond distance and all these things. Okay. And on the other end, for RT, what over HT, what we saw, though this proton is mostly residing in between the amide oxygen and the phosphorus oxygen. Okay, so what we did a database study. So there is actually more than 20 the agglomeratin salt co-crystals are reported in the literature. So what we did a simple study. So we did the pk of all these things, and co by cn distance we have plotted. Okay, so if you can see this, that's a big indication, because if the proton oxygen gas get protonated, okay, then the resonance structure will be changed, and practically the co distance will be more for the salt as compared to the co-crystals. And of course the co distance sorry, cn distance will be bit less. Okay, so with that, so co by cn is a very good indicator to having a salt or co-crystal. So okay, now if you see that we have color coded, all the black dots are all the co-crystals already defined by the single crystal in the literature and also in the paper, although in the database. And all the red circles, red triangles are the salt. Okay, salt. So now you see, surprisingly, when the powder data whatever we saw, the both the structure was sitting same time, because atomic positions and atomic bond lengths and all this thing was not correct. But when we corrected the structures with the DFTD calculation, then we can see the difference. So what happened? RT forms is became a salt. Okay, and it's also sitting nicely with the cluster of other salts. There is no ambiguity, and at the same time HT forms is became in between. So that means in between. So that's what we used to call is a continuum. In a summary, so RT form is salt, whereas the HT form is in continuum. Okay, so that's also we have verified with the spectroscopy technique. So what we did, so we don't have the variable temperature for the SSNMR or the FTIR. So we compared with respect to pure agomelatine, and we can see that for the for practically for RT form, for FTIR, there is a low wave number shifts. Okay, that indicates the strong interactions. Okay, same time for the SSNMR, we also see the downfield shift of the C13, carbonyl C13, C13. Means amide carbonyl C13. Okay, with respect to agomelatine, that indicates that it is a salt. Okay, and when we simulated the SSNMR of the high temperature form, that is we could not collect the data, so it's sitting in between. That very strongly confirms that what we are what we are got it from the powder structure and followed by the computational simulation. It's a right frame. Okay, so further, what we look bit further also. So what we tried here, so we tried to see the possible mechanism of the solid state, mechanism of the solid state transition. So that can be can be called as a diffusion metastatistics like transitions. So what we have viewed, we have viewed the structure as a the tri-layer building blocks. Okay, so where you read this is the polar region of a phosphoric acid, and the agomelatine is the hydrogen bonded, and other two part is the hydrophobic thing. So when we measure all these things, we can see that along A and C, both the material get expanded. Okay, so as for example, along A, so it's a 19.8 to expanded to the 18.4. Okay, and same way in a C direction also expansion happen. Okay, but surprisingly, so when we look the other direction, that is the B axis, we saw that is nothing but the pi stacking of the aromatic regions. We saw a contraction of 13 to the 13.7. Okay, now this is nicely got verified by the image analysis of the hot stage microscopy images. So what we did, so first we simulate the crystal structure, morphology of the material, and here we can see that our B axis is the long axis. Okay, and accordingly we measured the whatever the crystal, there is no interference, and here nicely we can see that there is also certain contraction is happened. So it's called the negative thermal expansion. So what if the mechanism is proposed is strongly verified by the experimental evidence also. With that, okay, so then we can thought about the concerted pathways of this following could be following. So first could be a proton migration of protonated amide to the phosphoryl group. Okay, then the change of molecular structures of the ACL, that bond distance and Jordan angle. Okay, then next layer, so it's happened the changing the layer structures. Okay, and afterwards changing the consecutive layers. So what it's called practically is by layer transformation. Okay, and with that, so in summary it can be tell that so it is a more integrated than the ordered disorder transition, that is what is generally known for these proton transport materials. Okay, this type of solid state sol to co-crystal phase transition is very rare. Actually, we haven't found any examples. That's according to our knowledge, they may be the first example. Okay, and this pathway also quite unique. Okay. With this, so in this slide I would like to discuss about the kinetic interplay, kinetic and thermodynamic interplay. So we have calculated the computationally the energy difference between the RT form and HT form, that we got that RT form is thermodynamically more stable. That to bisect this origin of this thing, we did the hospital calculation and we understand the strong hydrogen bonding interaction is towards contributing more towards this higher energy. And then same time, so as we know, there is not the thermodynamics playing a role. So there is a energy difference of a means thermodynamic or of a kinetic energy barriers is playing a role. We have determined this number by DSC isothermal study. Okay, and please note that this is not in a schematic, it's not in a scale. Okay, and what we observe is only a 30 to 40 kilocalorie per mole. On the other hand, is a very recent example. They are working with a nicotine and fumaric acid co-crystals, and they observed a solid state phase transition, but via nucleation and growth mechanism, and their energy activation energy barrier was is a practically 10 times more. So all this is also, it supports that what we have discussed in a last slide. So it's a very low energetic transition. That's why it's not changing a crystal structure, not even the morphology also not getting any affected. Okay, so also this thing also we verified with some solution crystallization. So what we did, we have changed the phosphoric acid molecular ratio with the agomelatine, and what we saw with the increasing of phosphoric acid, we start to see the getting the more HT forms. Okay, that is not accessible at room temperature, but in solution crystallization we see it. Okay, what could be a possible inference? So that there is a complex dissociation behavior of phosphoric acid probably playing a role. Okay, with that I would like to summarize this thing. Okay, so this protocol about the this powder structure, crystal structure solved by powder, and the DFD2 calculation, which can discrimination between salt and co-crystal ambiguously. Okay, was verified with the help of the well-founded chemical crystallography and database analysis and spectroscopic study. So this protocol we are trying to generalize with so many other molecules in a pipeline in our company. Okay, and it's a mechanistic understanding of this unique metastatic like transformation was well supported by the negative thermal expansion along the axis. Okay, this observation demands that phase transition stimuli such as temperature and humidity need to be considered while predicting the assigning or predicting and assigning the pharmaceutical multi-component material as a co-crystal or salt. So it's not at all straightforward that by I am looking the delta pk and I'm saying I will be getting the salt and co-crystal. What Ramana, he also said that you will be waiting for the results. Okay, or he has to be means you have to put some more protocols to consider these things. Okay. And then systematic evaluation of dissociation behavior of API and conformer in selected solvent systems for better process control to generate a salt or co-crystal form exclusively. So if we understand the dissociation behaviors of API or conformer with a selective solvent, so our success rate to form this controlling this salt or co-crystal will be much more. Okay, so from all these things, so what we can understand, the scientific understanding without any control strategy, no use for crystal, no use for industry. We have to implement. Okay, so now that, so I am coming to the second case study. Okay, so this is it is a polymeric co-crystals of Dapson and well-known excipients PEG. Okay, so so why we are doing these things? Okay, the reasons are following. Okay, the role of polymer as a co-former is not a well explored area for pharmaceutical co-crystal. There are some isolated examples. Okay, but they are also calling as a inclusion complex, not as a co-crystal. But we feel that this our whatever this category we are getting is a bit unique. I'll try to show the results towards this. Okay, and second thing, what we believe, so polymeric excipients which can enhance the drug functions towards drug delivery is not only the dissolution control. Okay, not to enhance, it can control. Okay, it can it can be a specific site specific, so many things can be possible because polymer can play a big role. So it can be a very much advanced co-former as compared to the small molecular co-former. So with this thought, so we did some study. Okay, for the Dapson, we are trying to co-crystallize with a 7 different grade of PEG, starting with a low molecular weight 600 to 35,000 molecular weights. Okay, and we standardize our protocol with a 6K. So first we did a phase diagram. Okay, so phase diagram, what we can see, so at the beginning up to 50%, so we can see the presence of new form as well as the PEG, and in above that we can see the binary phase of a new form and the PEG, but around 55 we see a pure phase. Okay, that we have characterized by the DSC. So you can see that this is the starting PEG melting point 63, Dapson melting point is 80. So it's coming mostly in between somewhere. So it's very surely confirmed that is a co-crystal forms. Okay, and if you see the powder XRD, so there is so many unique peak arise, and you can also note that low molecular weights or all the powder XRD pattern of different grades are mostly same. Okay, mostly same, only difference in a crystallinity. Okay, but the one of the challenge for the any crystal is determining the stoichiometry. Okay, this is not at all straightforward of a polymer excipients. Okay, so we did it a different way, and it's also it turns out right, that I'll explain. Okay, so we use a DSC tool, or DSC as a tool. We construct a again we make a binary mixture and construct, it's called the Tramans triangles, and that intercepts presents with a intercept of Dapson weight fraction with a enthalpy. They give the idea about the precise stoichiometry. So that weight stoichiometry, if we convert to the molecular stoichiometry, so we got 4:1 ethylene dioxide monomers. So one monomer versus one Dapsons. Okay, So also we did the IR to confirm that thing, and that makes sure that amine groups and the sulfur dioxide sulfur dioxide stretchings have changed. Okay, change that indicates there is some hydrogen bonding interactions happened. Okay, and then another extra step we did it. So we did the hot stage microscopy and also the variable temperature IR. That's a bit giving a bit uniqueness. So in a molten stage, even there is the hydrogen bonding interaction, there is a no change. That indicates in a molten stage also this material are still co-associated. That is quite different than the whatever is referred in the literature as a inclusion complex. In inclusion complex, what happens in general, the polymer get degrade or API get degrade, but this type of thing never happens. So that's why we believe it's a new category, new subset of a co-crystals. With that, just a minute. Okay, so now you can see that already I have discussed all the different grades. Okay, so having the same pattern that indicates all the structure is form a isomorphous structure, crystal structure, means their unit cell and the packing should be the same. Okay, and we are getting the better crystalline material with a 600 that we have indexed by powder XRD. We got a good cell dimension, and that gives us a strength to grow a single crystal, and luckily we grow a single crystal and we got a cell dimension and the stoichiometry and all these things that's exactly matching with our proposal whatever we got from the DSC. So it's a 4:1 PEG monomer versus 1 daption. Okay, and the crystal structure looks like that. So it's in general PEG having a 7:2 helical structure, but you can see that no, they have the twisted conformation, and this in that only the daption molecule inserted, and all the hydrogen bonding interaction what was seen in assigned in a IR, that's all are present here. So that gives a real strength to us that what it's all are started to collaborate, and what we understand that these strong NHO interactions, that is only the connecting part to daption to PEG. Okay, and this is a nice thing to share that this crystal structure was featured in 2018 by CCD website as a very first entry of a drug co-crystal with the organic polymer, because here we feel really extremely lucky to have this thing, because polymer generally don't crystallize. Okay, it's very difficult to crystallize, and on top of that is we are making a co-crystals. Okay, so that's why probably they understood that thing and they acknowledge these things. Okay, and okay, now here you can see that modulation of physical property. So already from the data you saw that crystallinity got decreased. Okay, with a weight of molecular weight. Okay, and it's get a plateau after certain molecular weight. Okay, and also is reverse thing happen for the melting point. So melting point get increase and get a plateau. Okay, and it is also the reverse thing happen for the hygroscope water absorption. So it's a low molecular weight having a higher absorption property, and compared to the compared to the high molecular weight. Okay, and so also we saw a nice solubility difference. Okay, so with any media we got a more solubility of the solubility of dapson 600 co-crystal as compared to the dapson, but it's not a huge. Here only what we wanted to show that what we want to show that there is a strategy to fine tune the physicochemical property through controlling the crystallinity, hygroscopicity and solubility by selecting the appropriate molecular grade of a polymer. Okay, so practically what we are trying to say that once if you luckily or it's in a by chance if you get a polymer and drug, okay, then the other physical property just you have to change the grade of the thing and whatever you want, and also the dose control and everything can be in your hand then. Okay, so that's but you have to be lucky. That's I'll explain something so what we are getting nowadays. Okay, so this is a strategy can be applied to modify the drug
for altering a desired solid form with a transferable manufacturability. Okay, so yeah, so last for 2 years, so we have been working with several polymers with a different grades and the various API, and we are trying to understanding the generality of this category of material. So you are not that much super lucky or very bad luck. So at least we got a 4 systems we have discovered. So with two more APIs and 2 more polymers. Okay, and what is the more interesting thing? So that's all I want to show to you. That's the this polymer now start to play a dual role. It's not only co-former. It's a stabilizer for a nanostructure and the particle and the nanoparticle engineering. So this is see the AFM image of one of this material. So we can control the material in a nanomaterials as a nanomaterials and see that. So it's a distribution also quite good. Okay, and what is happening nicely, if you see 100 to 400 nanometers particles, there is a core cell. So core is nothing but the crystalline material and cell is the polymer. So in that way they get stabilized. Okay, and also if we put this type of material in an organic solvent, we start to get the organogels. Okay, that's the another way to control the drug delivery. Okay, so and what we are doing, so you can see the nice PLM images of this is liquid crystalline material, because that means they have a lamellar structure. That lamellar structure we can model by the small angle scattering data. Okay, and we can find out the what is the lamellar thickness of the co-crystal parts and the amorphous part. So in a principally, so nowadays we are working to develop a coarse grain molecular simulation to understand the eutectic like local structures and evolution of such nanostructures materials. Okay, with that I would like to summary of this thing. Okay, so these amorphous drug polymer co-crystal can be considered as a emerging subset of pharmaceutical co-crystal. Molecular insights and melting behavior distinctly demonstrated that these co-crystals have found owing to strong hydrogen bonding interaction, unlike crystalline inclusion complex. We have further established a strategy fine-tuning of pharmaceutical physiochemical property through controlling the crystallinity, hygroscopy, solubility by selecting the appropriate molecular weights and grade for the polymer. Okay, and this drug polymer can be extend the scope of a multi-components API as an innovative alternative as a nanostructure material. And with that, I would like to acknowledge my seniors like Atanu Sen Gupta, Kallan Raman, Srinivas Duryasamy, K. Vyash, and Hamid Vishesh. Okay, and I would like to thanks for your presence. Any questions I can.